implants and silicone guns just as a sealant. So the market of silicone has increased in the last decades, and the manufacture of silicone worldwide is per year about 10 to the 7th tons of silicone. And this is a huge amount of silicone which is released in the environment, not, of course, not completely, but there's different sources of silicones which end up into the environment. So the silicone-based patents have increased in the last years, and as you can see in 2020, so there's an important increase, and I think it's still increasing, which reveals the importance of silicone and siloxanes in our life. So what are siloxanes? Siloxanes are volatile compounds, usually known as methyl volatile siloxanes, and we find basically two different types of siloxanes. So cyclic methyl siloxanes and linear methyl siloxanes. Linear methyl siloxanes are just a linear molecule, and the cyclic methyl siloxanes has a cyclic structure, but they both have in common that the molecule contains silicon-oxygen bonds. And the point here is that these molecules have different radicals that can be methyl groups, both in cyclic and linear methyl siloxanes, phenyl groups, or vinyl groups, or can contain fluor, for instance, or other elements. So we can find many different types, and this is related to the application of these compounds. All of them have in common that are oily, colorless liquids. So at room temperature, they are liquid. They have extremely low water solubility, and this is one of the problems of biodegradation of siloxane. So the accessibility by microorganisms to these compounds. So the solubility is between 0.005 to up to 1.5 milligrams per liter, but it's usually low than 1. So there are many different types of compounds, so the range is this one, but it's usually low than 1. They have a high vapor pressure and high Henry constant low, which basically means that those compounds are volatile and they evaporate easily, and it's very difficult to keep these compounds in a water solution. And of course, bacteria are living in a water environment. So the problem here is, how do we transfer these compounds from a gas phase to the water, or at least to a biofilm where bacteria are living in? Well, the siloxanes are considered as emerging contaminants because in the last decades the consciousness of exposure to siloxanes has increased, and you can find in this publication in Chemiosphere how the perception of the safety of siloxanes has changed. So in the very beginning, where siloxanes were synthesized and proposed to be used, there were some toxicological studies which give the impression that those compounds were safe, were at least completely safe. But from the very beginning, and I'm not going to go into detail into it, some toxicological studies show that those compounds were to some extent toxic. The point is that to detect the toxicity, you have to inject those compounds into the body, and those compounds are very volatile. So the exposure, the environmental exposure to these compounds is quite low. Anyway, those compounds were classified as safe compounds. Later on, they started to be used in cosmetics, in breast implants, and the effects, the side effects on breast implantation was observed, and this promote the revelation of these compounds. And in this fourth period, the toxicological studies were performed, so in a long-term period, and the scientists were aware of the necessity to monitor these compounds in the environment. In this last period, siloxanes were commonly identified in environmental samples, in water, in sediments, in soils, and this represented a breakthrough in defining environment as a source of human exposure to siloxanes. And this forced to limit the production of siloxanes. For instance, England reduced the production of siloxanes for some uses, and many many governments presented environmental reports about the siloxane as a pollutant. We can find publications worldwide showing the persistence of siloxanes in the air, in metropolitan cities, in the big cities, in different parts of the world. So the interesting point here is that we, during all these years from the 90s to up to present, many different types of bacteria have been described able to metabolize siloxanes, but there are no studies on how microbial communities in the natural environment deal with these compounds. So this is an open question. I think it's something interesting to be addressed, but it was not the focus of the thesis of Eliana. You can see here in this table, along these periods, how the interest in the impact of siloxanes in the environment increased, because the number of publications increased from the 90s up to now almost. Okay. The sources of siloxanes into different environmental compartments are summarized in this figure. I don't want to go in detail to explain it, but I think you can basically identify that the sources of siloxanes is just by plant industrial effluents that can act as emission of siloxanes in wastewater, but as well the use of personal care products. The consumption of these products can represent a source of siloxanes into the wastewater. And of course, those compounds volatilize into the air, and then from wastewater, they can go into the water environment, can be bioaccumulated in the biota, and can be absorbed into the sediment. And of course, wastewater biosolids can retain siloxanes, and then from the sewage can be transferred into the soil or into landfill. There are some publications which reported the bioaccumulation, the persistence, toxicity of siloxanes into the trophic web, and as well into in different carp species, in Cyprinus carpio, in China, or in the Korean Crucian carp, Carassius carassius. So I think this just shows us that siloxanes are considered as a contaminants that can be bioaccumulated in the trophic web, and I think that at least from the environmental point of view of microbial ecology should be something that we could focus. Okay, but the topic of the PhD thesis of Eliana was completely different, so it was not focused on the environmental in the environment. She addressed the question of problem, so the question to reduce siloxanes present in biogas. So you know that biogas is produced in wastewater treatment plants. That biogas is basically methane that is used as an energy source. Is not a fossil fuel energy. So the problem with this biogas is that it contains siloxanes, and once the biogas is used for combustion to generate energy, the siloxanes are converted in silicon dioxide. So siloxanes are transformed in silicon dioxide. These silicon dioxide are present in small particles that aggregate, they precipitate onto different types of machines that are using to generate energy. And then what we see is the presence of this white precipitate, which causes corrosion. And then, of course, this reduces the lifetime of these machines. So from the wastewater treatment plant requirements or interests, people address the question to reduce, how to efficiently reduce the amount of siloxanes into the biogas. And one way to do that is just using bacteria. But there are many different technologies that we can use to reduce the amount of siloxanes in the biogas. None of them have proven to be ideal. So some have some advantages, some have some drawbacks. The most common technique is the absorption onto activated carbon. The problem is that we need to use high amounts of activated carbon. We need to regenerate activated carbon, and this has a high investment cost. Another technique is to absorb into acid or base solution. Those are physicochemical approaches. And the biological approach, the biotechnological approach, is using the biotrickling filter. In this biotrickling filter, what we have is a matrix, solid phase, where microorganisms are growing, and a liquid media recirculating through this solid matrix, and a gas stream containing siloxane, which diffuses along this filter. So microorganisms are in contact with water and as well with siloxane. This biotrickling filter is an interesting approach, but the removal efficiency is not higher than 70%. So physicochemical approaches allow to reduce up to 99%, 95%. And the biotechnological approach can only reduce up to 70%. But the environmental impact of this technique is much lower. So if we calculate the environmental impact, assuming the CO2 produced using these technologies, these are one order of magnitude higher than using this biotrickling filter, and as well the operating costs are lower. So one idea is that we could combine a biotechnological approach with a physicochemical approach. So maybe bacteria are not good enough by themselves to completely remove siloxanes, and this is more related to the mass transfer problem of siloxanes from the gas to liquid phase than it relies more on this than on the capacity of microorganisms to use those compounds, because we isolated, and this is what I'm going to show you, we isolated many strains able to grow on siloxane. So it's basically the problem of the mass transfer. So using a combined technology, we could reduce the operation costs and reduce the environmental impact and raise the removal efficiency or keep the removal efficiency up to 99%. So just to keep in mind the importance of siloxane removal in wastewater treatment plant, a wastewater treatment plant save between 2.6 5.7 euros per day per cubic meter of treated wastewater. So I don't know how many thousands of cubic meters is treating a wastewater treatment plant, but I think that are several. So I think it would represent a high amount of money. Okay, so which approach did we use? This is a work that we did in collaboration with Alekia. So Alekia was in charge, or they were involved in developing a biotreatment filter, and Eliana was focused on the isolation of bacterial strains to be tested on these biotreatment filters. But the idea is that we have a biotreatment filter, biogas is injected into the column, then we have a support matrix where microorganisms are growing, and then nutrients are recirculated along the filter. What happens here? So in this compartment we have a support solid surface with biofilm growing on top, and the compounds present in the gas phase are transferred on the surface of this matrix. And then microorganisms should be able to use the siloxanes as a carbon source. So as you can see here, we have no carbon. So this is just silicon and oxygen. But if you remember, most of siloxane contain methyl groups. So bacteria can use the methyl groups as a carbon source. And then they release silanidiols and formamide and other simple compounds. I have to mention that we didn't work with real biogas. So Eliana, in her PhD thesis, she was working all the time in using just a gas which contained siloxanes and as well other organic volatile compounds, but it was not real biogas. And I say this because biogas contain as well sulfide, which is corrosive and toxic, contains CO2, so the presence of other compounds in the biogas can difficult or can impose some restrictions to the growth of microorganisms. So at this point we just use gas which contained only selected volatile organic compounds. So in a more detailed way, we have the activated carbon, which would be the solid phase. And this activated carbon acts retaining the siloxane and has a catalytic activity. So can open these rings and convert the cyclic siloxane in a linear compound. And microorganisms can both use the cyclic or the linear molecules and then generate the biofilm. So if we use just siloxane as a carbon source in this biotreatment filter, and we do not add any supplement, any organic compound, then we can assume that microorganisms are using just siloxane for growth because they don't have any other carbon source. These are images, scanning microscopic images from this activated carbon, so you can appreciate that microorganisms are growing on top of this activated carbon, and then they colonize completely this surface. Okay, so the first point was to isolate microorganisms able to grow using siloxane. And what we did was to sample, to obtain raw anaerobic sludge from a wastewater treatment plant which had problems with siloxanes. So we had a high chance to find those microorganisms. And we started anaerobic enrichments in bottles without oxygen. And in these bottles, what we have is a liquid media. And as you can see, there's this black material which is activated carbon. So we supplement the siloxane into the enrichment as activated carbon saturated with siloxane. So what we did was take activated carbon was saturated with siloxane, and then we added the activated carbon into the enrichment. And we could know exactly how much siloxane was inside. Then we just add a dilution of the sample. We let it incubate for several months. We could identify biofilm on top of this activated carbon, and preliminary results show that the amount of siloxane extracted from this enrichment was much higher in the control, where no microorganisms were present, than in different types of enrichment. So this extracted means when we finish the incubation, we just centrifuge the enrichment, we remove all the activated carbon, and then we extract the siloxane. And then we can calculate how much siloxane is consumed. So the consumption was higher in the treatments with using microorganisms from the activated sludge. Then we got, so these results suggest that we have microorganisms which are active using this siloxane as a carbon source. Obviously, this raw sludge contains other organic compounds. So probably there was other organic compounds here, but we further isolate in Pew culture the microorganisms present in the enrichment. We isolated in agar plates containing only siloxane D4 as a carbon source. So those strains were growing in a mineral media only with siloxane D4 as a carbon source. Okay, overall we got about 50 bacterial isolates. These are the species that were identified. We identified the isolates by sequencing the 16S rRNA gene, and the most probable identification of the isolates is this one. So bacteria that belong to the genus Pseudomonas, Ciceribacter, Alicycliphilus, yeah, different species. What is interesting is that the species that were identified in the enrichments are different than the ones which were isolated directly from a biotrickling filter, which was already operating in the Lechia laboratory. So we did two different types of isolation. So we isolate microorganisms from the enrichments, and we isolate microorganisms from a biotrickling filter, which was supplemented with raw sludge. And what we got is different bacterial strains. This could be due because the inert support is different. So in a biotrickling filter was mainly composed by lava rock, and in the enrichments what we used is activated carbon. And this could be one of the reasons to obtain different bacterial species. So we compared the similarity in terms of 16S RNA gene similarity of these isolates with previously identified microorganisms, and some were close related to microorganisms able to use hydrocarbons and use simple organic compounds. And this suggested us that those microorganisms, beside use D4, could be maybe able to use other organic compounds like toluene, hexane, and other contaminants. Okay, one interesting point that we found is that the members of actinobacteria, so the isolates that were obtained from the biotrickling filter, those actinobacteria contain specific biosurfactants on the cell surface. So surfactants are molecules that contain a polar group. So you have here hydroxyl radicals, which can be ionized. So they are. This is a polar side, and this is a polar side, soluble in oil. And many different types of actinobacteria can produce these compounds. For instance, Mycobacterium tuberculosis or Rhodococcus erythropolis, which in fact we got one of these isolates. And the purpose to produce these compounds is that bacteria increase the availability of hydrocarbon molecules by generating these biosurfactants, because these biosurfactants interact with an hydrocarbon molecule or a molecule with a low solubility in water, and then microorganisms can better access to these compounds. Once we isolated these different bacterial strains, we selected some of those strains to check the efficiency of the strains growing using siloxane as a unique carbon source. And we realized that methylidium isolate 58 reach higher optical densities, and Pseudomonas as well. So basically these two were the best candidates to further explore their capacity to be used in a biotrickling filter. Other isolates just reach very low optical density. So the biomass which is generated using only D4 as a carbon source is very low. So bacteria do not reach high optical densities. Usually when you use rich media, you can easily reach to 0.9 units. And as you can see, the growth just finished at about optical density of 0.4. Okay, then what we did with the isolates is we selected methylidium and Pseudomonas because they were the most promising candidates, and we studied the capacity of these isolates to grow to use D4 as a carbon source, just calculating the removal efficiency. So how much siloxane can be removed from the media. And the strains were growing only with D4 or using D4 together with other volatile compounds, because this is a more realistic condition. In the biogas we have many different other carbon compounds, not only D4. So what was interesting for us is to check how bacteria could grow using siloxane, D4 and D5, and as well other organic volatile compounds, in this case hexane, toluene and lemon. And what is interesting is that when bacteria, methylidium and Pseudomonas, is growing only with D4, the removal efficiency is higher, but when they grow in a mixture with other compounds, the removal efficiency decreases because they have probably more preference for these compounds than for siloxane. But when they grow in co-culture, what we did is to use a selection of different isolates in a co-culture condition, simulating a microbial community. And in this community, we have both methylidium and Pseudomonas. In this case, the use of D4 was decreased, But when bacteria were growing in presence of these volatile compounds, the consumption of D4 was increased. So this is an opposite trend compared to what was observed using only methylidium and pseudomonads. And this could be explained by maybe one reason is that the presence of other volatile compounds can stimulate the pathway of siloxane biodegradation. So in absence of these compounds, bacterial interactions do not promote siloxane biodegradation, but in the presence of other volatile compounds, biodegradation of D4 is stimulated. Okay, next step was to use methylidium in a biotrickling filter. This was the main goal. If we have a bacteria which is able to metabolize D4 in a batch experiment, then what we want to do is to use this microorganism in a biotrickling filter and calculate how much siloxane can be removed. This is basically the design of the biotrickling filter, and like I said before, we have a liquid media, recirculating, and a gas stream which is injected into the column, and then what we did is to measure the D4, the difference of D4 which is injected and the difference and the D4 which is released. It's basically this. Removal efficiency is the difference of concentration of D4 at the beginning, at the end, divided by the initial concentration, and then we got the removal efficiency. So I'm going to show you now in the next slide the results of removal efficiency, the elimination capacity. This is another way to calculate the efficiency to remove siloxanes, and this applied to the volume of the bioreactor and the gas stream. Basically, what we did was, the aim was to reduce the size of the biotrickling filter. One of the limitations in siloxane removal is that the biotrickling filters need to be very big, and then these imply high amounts of packing material, high amounts of lava rock, and activated carbon. So one point was to reduce the size of the bioreactor. In a stage 1, the size of the packing material was 36 centimeters, and the empty bed residence time was 15.5 minutes. This time is the time of gas contact with the solid material. So if we reduce the size of the filter, then we reduce the contact time. And the idea is to find an ideal size of the filter, an ideal empty-bed contact time in order to maximize the siloxane biodegradation. Okay, so in these conditions, what we have is that along the different days of biotrickling operation, we have here both the loaded compounds, hexane, toluene, and limonene, and the elimination capacity. The load compound is the line, and the elimination capacity is the dot. So when the dot matches the line, we have 100% biodegradation, because line means the amount which is loaded, and the dot means the amount which is degraded. Okay, so for toluene, for limonene, the biodegradation was completely, but not for hexane. So this is the elimination capacity of hexane, and this was the amount which was loaded. So it was really far from so a lot of hexane was not biodegradable. For siloxanes, it was a similar trend. So this is the amount which was loaded before D5, and this was the amount which was removed. So at least for D5 was about 50% in the best case. In the stage 2, The size was reduced with a reduction of the empty bed contact time, because if we reduce the size, then the contact time is reduced as well. And here what we can see is that hexane removal was increased. And then we did here a change. In this condition, we made the change. It was just to supplement the liquid media with higher nitrate concentration. So the liquid growth media contains nitrate at 1 gram per liter. So we just increased to 2.5 grams per liter. And then the consequence was to increase the hexane removal. So methylidium probably can use nitrate as an electron acceptor, and this probably promotes the hexane biodegradation. So these results, what show us is that using this bacterial strain and just changing the growing conditions, we could favor the biodegradation of specific volatile compounds. For D4 and D5, the biodegradation of D5 was increased as well using a higher nitrate concentration. And in stage 3, the size of the treatment filter was still decreased. The empty bed contact time was decreased as well. And in this condition, what we tested is to add 1% oxygen concentration, because biogas contain traces of oxygen. So we thought that maybe the addition of oxygen could stimulate the removal of siloxanes or other volatile compounds. And for the hexane was the case, not so efficient than using nitrate. But we could not get a better result for D4 and D5. So for D4 and D5, basically the main problem is the mass transfer. So the capacity to increase the transfer of D4, D5 from the gas phase to the liquid media. So finally, besides on working with few cultures like methylidium or pseudomonas, a good approach can be to use complex microbial communities in a biotrickling filter to study siloxane biodegradation. So not using one bacterial species or a combination of few bacterial species, but supplementing the lava rock directly with activated sludge, which contains high amount of microorganisms. Then, if we just operate this biotrickling filter for several months, we assume that using only D4, siloxane D4 or D5 as a carbon source, then we could get a selection of microorganisms able to use these carbon sources. I didn't mention before that these biotrickling filters were operated always first abiotically to check that a siloxane was not removed. So always for any study, the biotrickling filter was working. So the siloxane, in all the experiments, siloxane concentration without bacteria at the beginning, so the entrance, in the inlet and outlet, the concentrations were more or less the same. So the absence of abiotic biodegradation was always checked. Okay, so we took activated sludge to study the biodegradation in a biotrickling filter during about 7 months. In this graph, what you have is the different stages of operation, and these different stages are defined by the addition of only D4 as a carbon source, stage 2, the addition of other volatile organic compounds, D5, hexane, toluene, and limonene, and using lava rock as a packing material. Then in stage 3, we added a supplement of activated carbon. And then in a stage 4, we use only activated carbon as a packing material, just to check for differences in microbial community composition and siloxane biodegradation. And all the changes were as well related to change into the empty bed residence time. So in this case, the empty bed residence time didn't change, and then we decreased the empty bed residence time to check whether siloxanes could be better biodegraded, but it was not the case. But what is interesting to remark here is that the removal efficiency is always very high for limonene and for toluene. So those compounds are easily biodegraded by microorganisms. But for D4, D5, D4, these are the two types of siloxanes that we used, and as well for hexane, the biodegradation, the removal efficiency was much lower. What is interesting to remark here is that the removal efficiency was increased by adding the activated carbon, at least in this period. These are the values of removal efficiency that were achieved. Okay, so we analyzed the microbial community composition in the different operational stages of the biotrickling filter. This is the sample code from the very beginning during all the time course, the sampling day. These are the number of 16S rRNA gene sequences that were obtained. The number of O2s, operational taxonomic units, good coverage, which tell us how many O2s we are able to recover from the natural community, and the Simpson index of diversity. And what is interesting here to show is that we detected a reduction in the number of O2s along the incubation period, which was due to a specialization of the microbial community to grow using these compounds. And the Simpson index of diversity decreased as well from close to 1 at the very beginning, because at this sampling day what we had here was the natural community from the sewage sludge, but then the diversity was decreased. What is interesting to point is that when we made some change here in the operational conditions, we could identify a change in the diversity. For instance, when we use a multi-component mixture here, we could check a reduction of the diversity, or at least all the number of O2s drastically decreased in this sample. Okay, which type of microorganisms did we identify? This is the microbial community composition, which are the orders that were identified, accounting for more than 1% of the total sequences. So if we compare the original sample, the composition of the original sample, to the composition of the different samples along the operational conditions, we can realize that there's a selection of specific groups. For instance, this group was selected as a dominant group from the very beginning, and some bacterial groups were detected consistently along the operational conditions. There are some changes in the composition that we can identify. For instance, when we added different compounds, so this is only using D4 as a carbon source, but when we added other organic compounds, the composition of the community changed. But then it was to some extent recovered to the original community that was identified. This is a change. There's another change here. This was related to a change in the hydraulic retention time. So it was changed, the hydraulic retention time was changed here, was lower than in these conditions, and probably this is at least the only explanation we have for this change. These are the main groups that were identified, and the isolates that we initially obtained from the sewage sludge, they belong to these groups. So it's not strange that we identify these orders since the isolates belong to these phylogenetic groups. Then an interesting point was to test if we could identify significant differences in microbial community composition, comparing the packing material without or with activated carbon. So absence of activated carbon means just lava rock without activated carbon. Presence of activated carbon means lava rock plus activated carbon. And we did a principal component analysis, and what we got is this sample distribution, but we could not confirm that this distribution was significant. So just doing for instance, Permanova analysis, we couldn't find significant differences in this distribution. And one reason could be that what we got in the biotrickling filter was a microbial community developed on top of lava rock at this point, and then when we added activated carbon, this activated carbon was colonized by microorganisms already present in the lava rock. So if there's an efficient colonization, those microbial communities would not be, to some extent, very, very different. At least this is the conclusion that we got. Okay, so in this slide, I show you the main orders that were identified. But the question was, can we identify microorganisms that are consistently present along all the bioreactor performance? So could we identify a core microbial community? Core microbial community is composed by microorganisms which are consistently identified in different samples that are suspected to play a key role in this community. Like for instance, in this case, for siloxane biodegradation. So we could identify O2s which accounted for more than 55% of total sequences. 35 O2s were always present in all the samples recovered, and those O2s accounted for, in this case, in the beginning, 60, 63, 69% of the sequences, and to some extent to 78, about 80% of the sequences. So we look at these microorganisms, for instance, members of Rhizobiales and Corynebacteriales, which increase at the later steps of the operational conditions, and some they were decreased. So looking at more detail to these O2s, which were dominant, they are close related to isolated strains that we got in the beginning of our study. So what is enriched in the biotrickling filter are microorganisms phylogenetically similar to what we got in our enrichment experiments. For instance, close to Methylidium, close to Allicitolithilus denitrificans, close to Pseudomonas, or close to Nocardioides. ISO means isolate, so the isolates we got. O2 means an identified 16S RNA sequence from the bioreactor. And these sequences were closely related to bacteria described with a capacity of to use single carbon compounds like methylamines, silicon carbon compounds like methyl siloxanes and a variety of other organic compounds, limonene, monoterpenes, all these which are listed here. So what we got is a phylogenetic and functional redundancy for the biodegradation of siloxanes and other volatile organic compounds. So the idea is that what we got in this biotriggering filter was a specialized community that could remove siloxane, but as well other organic compounds. So this could be a source of functional diversity that could be further explored. And just to conclude, the final remarks are that the organosilicon compounds are considered emerging environmental pollution that cause a negative impact in the wastewater facilities, so both as emerging environmental pollutants and as compounds that cause these negative impacts. And promising bacterial isolates can grow using only these compounds as a carbon source and can be used for biogas upgrading in biotechnological fields. Co-cultures, double siloxane removal in presence of other volatile organic compounds, suggesting that microbial interactions favor siloxane biodegradation. Combination support of super materials like lava rock and activated carbon increase siloxane removal and promote the selection of different bacterial microbial communities, although no significant differences were observed. A core microbial community harbors phylogenetical and functional redundancy, and suggesting that robust community, suggesting the presence of a robust community to disturbance operating changes in the biotechnological field. Best removal efficiencies in the biotechnological filter for D4 and D5 were about 40% and 45% respectively. So those are not very different to what was observed in the batch experiments. So we were not able to more efficiently decrease this siloxane removal. And finally, the hybrid technologies combining biological process and absorption or new tailored absorbents offers a green solution for siloxane removal. So still, we think that there is still many aspects that could be improved in order to increase this removal efficiency, and we still believe that these hybrid technologies can offer a solution. And finally, I would like to thank to all the people who was involved in the PhD of Elena Boada Caguanyes. So especially to Alba Cabrera Cudony and Eric Santos from the Lequia, who performed all the biotriggering filter setup and all the analysis on siloxanes, and to Luis Bañeres and Maria Martin. So that's it. So, do you have any question?